Right. Good morning, everyone, or afternoon, wherever you are in the world. Welcome to the session on prevention. I'm Gilda Tastron from the Burnett Institute in Melbourne, Australia, and my co-chair is Jim Turpin from NIAID, NIH.
Good afternoon, everyone.
Yep. We have a great lineup of speakers for this afternoon, starting with a presentation on a live biotherapeutic approach for preventing bacterial vaginosis recurrence. We will then shift gears from the vaginal microbiome to the penile microbiome and learn about its role in mediating immunological changes that modulate HIV susceptibility. We then have two short talks delivered by graduate students focusing on the metabolites produced by the vaginal microbiota and their role in HIV replication and cervicovaginal epithelial barrier function. So all speakers will respond to the questions at the end of this session, so please feel free to type your questions in the chat box. So it's my great pleasure to introduce our first speaker, Dr. Laurel Lagenaur from NIH and OSEL in the United States. Laurel is a molecular biologist with extensive experience in virology and bacteriology. And as Director of Research at OSEL, which is a small biotech company, she developed a recombinant lactobacillus technology platform, leading to the development of the novel topical microbicide for women called Mucocept HIV, which is Lactobacillus jensennii, engineered to express a potent HIV entry inhibitor. But today, Laurel will be presenting data on another live biotherapeutic called LACTIN-V, which is Lactobacillus crispatus, for preventing recurrence of bacterial vaginoses and ultimately HIV acquisition. The title of Laurel's presentation is Connecting the Dots, Translating the Vaginal Microbiome into a Drug. Over to you, Laurel, and it's absolutely great to see you again.
Good day. I'm Laurel Lagenaur, and my presentation is called Connecting the Dots, Translating the Microbiome into a Drug. I'd like to thank the organizers for inviting me to give this talk. My conflict of interest: I'm an employee and stockholder at OSEL, the company that's developing LACTIN-V. Bacterial vaginosis is a dysbiosis of the vaginal microbiome. The optimal vaginal microbiome is low diversity and lactobacillus dominant. And whether you look at a gram stain or a bacterial communities plot shown here, you can see that low diversity of mostly lactobacillus. Conversely, the pathophysiology of BV, you actually see a decrease in those hydrogen peroxide producing lactobacilli. And again, the gram stain and the bacterial communities plot paint a very different picture. We see increased bacterial diversity. This can be accompanied by, it's accompanied by high pH and can have odor and discharge, although bacterial vaginosis may be asymptomatic. Doug Kwong's group at the Reagan Institute found that lactobacillus deficient cervical bacterial communities are associated with increased HIV acquisition in young South African women. In this study, they found that women with lactobacillus dominant cervical types had the lowest levels of pro-inflammatory cytokines and the lowest risk for HIV infection. Conversely, women with a diverse microbiomes, shown here on the right, cervical types, had the highest levels of pro-inflammatory cytokines, up to 17-fold increase in activated CD4 T cells, the T cells that would be HIV targets, and about a 4-fold more increase likelihood of becoming infected with HIV. So our hypothesis was that lactobacillus dosing, Lactobacillus crispatus in particular dosing, following the standard of care antibiotic treatment for recurrent BV, could also reduce the susceptibility to HIV by creating a lactobacillus dominant cervicovaginal environment. So our product is LACTIN-V. It is a single strain of Lactobacillus crispatus, CTB05. It's delivered as a powder in a vaginal applicator. In phase 1 studies, it was safe and well tolerated. The phase 2 studies, which I'll talk about today, were sponsored by NIAID and DMID. LACTIN-V targets the underlying cause of dysbiosis, which is a loss of those lactobacilli, and it's an adjunct therapy following antibiotics. The phase 2 clinical trial has already been completed, but if I have time at the end of the study, I'll talk about another trial that's in progress to look at colonization reduction of genital tract inflammation and susceptibility in South African women with HIV, with high risk for HIV. So this is the phase 2 LACTIN-V clinical trial outline. OSEL, in collaboration with 4 centers, performed a double-blinded, placebo-controlled phase 2b clinical trial to prevent bacterial vaginosis in women treated for BV with metronidazole gel. Women ages 18 to 45 were screened for eligibility, including a BV diagnosis. Women that completed a course of antibiotics, if then they were eligible, they were enrolled at a 2-to-1 ratio to receive LACTIN-V or placebo for 5 days, shown here, followed by twice weekly for 10 weeks. Women were followed up at 4, 8, 12, and post-dosing at week 24. And the primary outcome of the study was women with recurrent BV at week 12. Here's the demographic data of the study. Most of the women, 51%, had greater than or equal to 5 BV episodes. 15 or 16% had 3 to 4 BV episodes. 20% had 1 to 2 episodes, and only 5% were having their first episode of BV. Adherence to the study was about 80%. It's important to note in the demographics that 20% of the women were Hispanic or Latina, and 42% Black or African American, making over 50% of the women in the study in the 2 populations that are most susceptible to BV. These are the criteria used for BV diagnostic. The clinical criteria of AMSELS, which looks for homogeneous thin discharge, clue cells upon a microscopic exam, increased pH greater than 4.5, and a fishy odor caused by amines from the bacteria associated with BV. And they also used a gram stain or Nugent's criteria, which looks for lactobacillus or other morphotypes. And women were enrolled if they had an AMSEL of 3 or 4 and a Nugent's of 7 to 10. These are the results of the phase 2 trial. In the intent to treat analysis, BV recurrence through the 12-week visit was significantly less in the LACTIN-V arm, 30% versus 45% for placebo, with an odds ratio of 0.54 and statistically significant at 0.031. BV recurrence was also significantly less at week 24, 39% in the LACTIN-V group versus 54% in the placebo group. Again, an odds ratio of 0.54 and a p-value of 0.03. At 12 weeks, Lactobacillus crispatus was detected in 79% of women in the LACTIN-V arm, and what was detected was CTV05, and 3% in the placebo arm, and this was another Lactobacillus crispatus, not CTV05. And this suggests that you need to give exogenous lactobacillus to increase those Lactobacillus crispatus numbers in women. So these are participants in the LACTIN-V arm. Here we have detectable colonization on the y-axis, the study week of visit on the x-axis, and in the second y-axis we have the concentration of lactobacillus. The blue box and whiskers plots show the median concentration of lactobacillus in log 10 CFU per mil. And you can see that all of the women in the study that were treated with crispatus that colonized had greater than or equal to 10 to the 6 CFUs. The gray bars are the proportion of women with detectable CTV05 per week visit. So you can see we were above 80% the first few weeks, and that drops to about 50% at week 24. Another way to look at this is the concentration of lactobacillus in participants who were treated with CTV-05. Here are women who were BV negative. You can see that they have high levels of crispatus, mostly over 10 to the 6th, and women who were BV positive have levels less than 10 to the 6th. So that suggests that these higher levels were protective. This is week 12 and week 24, and you can see those levels stay high in women that maintained lactobacillus colonization. When we looked at colonization stratified by sex or menses, we could see that whether women had condomless sex or sex with a condom, the levels of lactobacillus at week 4, 8, 12, and 24 were the same for the two groups. This is women with menses and without menses, again, week 4, 8, 12, and 24. So neither of these situations negatively impacted lactobacillus colonization. Regarding adverse events, all the adverse events were mild or moderate and were equally distributed between the LACTIN-V group and the placebo group, and there were no severe adverse events in either group. This is a qPCR of women that were successfully treated with LACTIN-V. We can see the copies per mil here on the y-axis and pH on the secondary y-axis. So we can see that these women start out with no levels of crispatus or CTV-05 shown in the purple and dark purple bar lines. When women were successfully colonized, we can see that all of the qPCR values, whether total bacteria, lactobacillus, crispatus, or CTV-05, track together. And we can see that pH starts out above 5 and drops to 4 in both these women when they're successfully treated with LACTIN-V. Now, here is a woman on the left that was unsuccessfully treated. You can see her lactobacillus levels go up, but they start to decline by week 12 when the dosing ends. And we can see that her pH went down, but then it comes up as lactobacillus crispatus goes away. And here we have another lactobacillus, maybe Eners, but you can see there's probably a log more of BV bacteria. So this woman's BV returned. And this is a woman who was treated with placebo. So you can see that she never had lactobacillus crispatus and she never had LACTIN-V. And you can see she had another lactobacillus that goes up and down, but her pH is very irregular and is high. And again, there's high levels of probably BV bacteria. So in summary of the phase 2B LACTIN-V trial, CTV-05 was detectable at protective levels through the 12-week dosing and at 24-week follow-up. Protective CTV-05 correlated with a 46% reduction in bacterial vaginosis recurrence. Lactobacillus crispatus levels in placebo arm remain low. They were about 3% versus 79% in the LACTIN-V arm, confirming the need to supplement with exogenous lactobacillus crispatus. No participant had a severe or serious AE, and LACTIN-V following vaginal metronidazole gel treatment significantly reduced BV recurrence and enhanced lactobacillus crispatus colonization, and thus had a major impact on the vaginal microbiota towards its optimal state. Some of our proposed next steps: we are working with the Chan Zuckerberg BioHub to do microbiome analysis on the women. These are some very preliminary results. These are women that had BV cures in the LACTIN-V and placebo group. Lactobacillus crispatus is the yellow set of bars. So you can see many of these women start out with high levels of BV type bacteria, Gardnerella, Smithia, Prevotella in these dark purple bars. and then they transitioned to Lactobacillus crispatus. This woman was considered a BV cure, although it looks like there may be some BV bugs at week 7. Converse that to the placebo arm, where you see only one woman obtained crispatus during that time, and by week 7, most of her crispatus was gone. Most of the other women reverted to Lactobacillus iners, which is this light green bar, or other lactobacilli. Some additional proposed next steps are to complete and submit our CSC to the FDA and to the Division of Microbiology and Infectious Disease. We'll have discussions with the FDA about a single phase 3 trial with the addition of this phase 2B trial for a new drug application. And we're planning and financing the phase 3 trial to have about 400 to 500 participants at about 10 clinical centers. I'd like to acknowledge all my colleagues who worked very hard in the study. Craig Cohen, pictured here, who ran the study. His right arm, Anke Hemmerling, groups at the STAR STI clinical trials group, Emmis, who did all the statistical analysis, Rupert Kaul, who's doing cytokine analysis, the Chan Zuckerberg BioHub, and of course the participants who helped out in this study. And that's the end of my talk, and I'd like to thank you very much for listening. I'll take questions.
So thank you very much, Laurel, for your presentation. Very interesting presentation. We will take questions at the end of the session. So I would now like to introduce Dr. Rupert Kaul, who will do the impact of penile microbiome on local immunity, immunology, and HIV susceptibility. One of the few of us out there who pays attention to the penile microbiome and really looks at it in a very interesting and focused manner. Rupert is got his clinical training in infectious diseases at the University of Toronto, and was a research fellow at the universities of Nairobi and Oxford for several years, where he completed an immunology PhD. He returned to the University of Toronto and University Health Network, where he now currently serves as director of clinical division of infectious disease and runs a clinical research lab focused on interactions between HIV transmission, genital and rectal immunology, sexually transmitted infections, and the microbiome. He works with cohorts in Canada, Kenya, and Uganda, and with the support of the University of Toronto and the OHT and endowed chair in HIV research. He was elected to the American Society for Clinical Investigation in 2013. So Rupert, take it away.
Hello, everybody, and good morning. My name is Dr. Rupert Kaul. I'm going to be talking to you today about the impact of the penile microbiome on immunology and HIV susceptibility. I really want to thank the organizers of the International Workshop on the Microbiome and HIV for inviting me to speak. This is a fantastic meeting, and I hope everybody's enjoying the speakers so far. I don't have any conflicts of interest, but I do want right up front to acknowledge several critical collaborators, and I'll tell you more about them at the end, but Dr. Ronald Galawango, Dr. Cindy Liu, and Dr. Jessica Proja. Any one of the 4 of us could have presented this work. And thank you very much for inviting me to present at this time. The work that I'm going to present is based in 2 large cohorts from Uganda, the Rakai Community Cohort Study of over 20,000 individuals in quite rural parts of Uganda, but with relatively limited immunology infrastructure. But this is the site where one of the randomized clinical trials of circumcision was performed that showed the reduction of about 60% in HIV risk. And IRV-UVRI, where we have fantastic vaccine immunology facilities and a great immunology infrastructure, as well as links to clinical cohorts in the Entebbe community. So I'm going to address these 2 questions. How does the foreskin increase HIV risk, and are there non-surgical alternatives to penile circumcision, since many men don't want to undergo a surgical procedure to reduce their risk? And I'm going to be emphasizing 2 things. I'm going to be emphasizing the integrity of the barrier, which is important in terms of HIV acquisition at any mucosal site, This cartoon here from Dr. Fauci's group appears to be showing a cartoon of the gut mucosa, but of particular importance, and something that's been a focus for our group, is looking at the density in the underlying tissue of activated CD4 T cells as HIV targets. So that virus has to first cross the barrier and then be able to infect activated cells and generate productive infection in that way. I'm not going to go in great detail into the background of the penile immunology studies that we've done, but Dr. Proja, during her PhD work and her subsequent work at the University of Western Ontario, has shown several key things. One is that IL-8 levels in the coronal sulcus, so that is the space with very low oxygen density deep to the inner foreskin, is enriched for IL-8. So this is by far the most abundant cytokine in the coronal sulcus, and IL-8 levels within that coronal sulcus correlate with CD4 density in the foreskin tissue that is overlying that microbiome, that space, and we'll see later that that's related to the microbiome within the coronal sulcus. The adjusted odds ratio for HIV acquisition in men who do have detectable IL-8 levels in the coronal sulcus is about 2 or 3-fold increased risk. And the foreskin tissues themselves are enriched for a number of key HIV target cells, both CCR5 positive CD4 T cells, and in particular Th17 cells that both myself and Lyle McKinnon and Tom Hope have shown are key HIV target cells very early in the process of HIV acquisition. Jessica showed in her work that those Th17 cells are decreased in the foreskin of men who are exposed to HIV without infection, and increased in the context of men who are herpes 2 seropositive, which is known to be a risk for HIV acquisition. So what induces that local cytokine production? We know there's a precedent in women where there have been many studies looking at the genital microbiome, inflammation, and HIV acquisition, that the presence of certain bacteria in the microbiome, which tends to divide itself very nicely into different community state types, either predominated by a lactobacillus species or predominated by diverse gram-positive and gram-negative anaerobes. The density of certain bacteria is clearly associated with more inflammation, and certain bacteria clearly associated with less inflammation. And that's shown nicely in some work from Brett Shannon in our group, with the x-axis showing the bacterial density in the female genital tract microbiome, the absolute abundance of different bacterial species, and the y-axis in each of these figures showing the proportion of women who have genital inflammation defined by cytokine levels. And what you see is that for Lactobacillus crispatus, an increased density of Lactobacillus crispatus is associated with a lack of inflammation, whereas Lactobacillus iners was neutral in terms of genital tract inflammation, and Prevotella bivia in particular, but Gardnerella vaginalis as well, is strongly associated with increased risk of inflammation as we have an increased density of that bacterial species. So this led us to be interested in the microbiome in the foreskin, specifically the coronal sulcus, and its association with inflammation. We know that penile circumcision dramatically changes the microbiome. This is showing how the shared microbiome that's very similar within the clinical trial at baseline prior to circumcision within Ugandan men diverges dramatically after circumcision, with a loss of gram-negative and gram-positive anaerobes, and a gain of skin-type bacteria, but particularly Corynebacterium species. In the initial work, we saw reductions, and this is Cindy Liu's work. This initial study showed reductions in 10 specific bacterial genera after circumcision, and our initial immune analysis focused specifically on those 10 genera that we had already shown to be decreased after circumcision. And if you look just at those in the men who remain uncircumcised within the clinical trial of circumcision, you'll see that an increased density of almost all those bacteria is associated with an increased odds ratio of HIV acquisition, with one exception, and that exception is a negative Negativococcus. So one bacterial species, interestingly, was not associated with HIV acquisition among those men who remained uncircumcised. But in those who remained uncircumcised, all these other ones that decrease after circumcision are associated with an increased risk of HIV acquisition. we were interested in looking at whether that related to the same immune parameters that Jessica had already shown were linked to HIV acquisition. And the answer is yes. So that as the density of Prevotella increases, when you hit an inflection point, when you hit a certain density of Prevotella, we then see a clear linear increase in the levels of IL-8. Same thing for Porphyromonas, same thing for Feingoldea. And interestingly, the only exception that was not associated with inflammation at any density was Negativococcus, that same bacteria that was not linked to HIV risk among those men who remained uncircumcised. Cindy has now gone on to do, over the last year, a much more in-depth analysis of the penile microbiome and its association with both immune parameters and HIV risk. This work's unpublished, and so thank you very much to Cindy for letting me present it. I think the first main message is that unlike the vagina, and very interesting to me anyway, there are no clear community state types within the penile microbiome. Most men have the same core penile bacteria, just present in differing amounts. 6 specific bacterial species, however, the bacteria tend to travel in packs. So as the density of one goes up, the density of other species go up. But 6 specific species were associated with each of HIV risk, the levels of IL-8 in the foreskin, and the density of CD4 T cells expressing CCR5, so those target cells in the underlying foreskin tissue. We call those basic species or bacteria associated with seroconversion, immunology, and cells. And those include Peptostreptococcus, anaerobius, 2 species of Prevotella, including Prevotella bivia, and 3 species of Dialista, again, bacteria that have been linked to bacterial vaginosis and inflammation in the female genital tract. Interestingly, no foreskin bacteria protected against HIV. Corynebacterium was associated with reduced levels of IL-8, but we did not see anything protecting against HIV acquisition. And Lactobacillus crispatus, that's protective in women, actually was associated with increased levels of IL-8 and an increased risk of HIV acquisition. If you do a network analysis, which Cindy has done with Dan Park and her group, what you see is that you see 2 clusters of bacteria, not just in the seroconversion cohort, but this is reproduced in a completely independent cohort of men where we ran the cellular immunology studies. And what you see is that in one cluster are the bacteria associated with increased levels of IL-8. So an orange or a red color means increased levels of IL-8. So those are the basic species in particular. Whereas in a completely separate cluster, we see Negativococcus, Corynebacterium, and Staphylococcus associated with a decreased level of IL-8, indicated by the blue coloration here in this figure. And if you then look at the immune parameters in more detail, both IL-8, and I'll show you the cell density afterwards, you see that the density of bacteria is not what drives inflammation. So here we have 2 groups of men in the left-hand figure. One has a high density of basic species, the inflammatory species. One has an equivalently high density of bacteria, but it's the control species. And we see the same density, but very different levels of IL-8 between these 2 groups. And we also see similarly low levels of IL-8 in men with an overall low density of bacteria, including a low density of the basic species. Particularly interesting to me is looking at the cellular immunology in terms of associations with the microbiome. So again here, Dr. Proja, Jessica, has looked at the group that have the high density of basic bacteria, the high density of the control bacteria, and then low density of all bacteria, including the basic species. And what you see is that that microbiome of the coronal sulcus is associated with dramatic increase in T cell density within the inner foreskin. So the high basic bacteria associate with the very high density of bacteria of T cells within the inner foreskin, not within the control taxa, and not the low density bacteria, and not at all with a T cell density in the outer foreskin, which is what you would expect, because of course the outer foreskin tissues are not in contact with the coronal sulcus microbiome. Interestingly, there's also no impact on proportion of CD4 T cells within those T cells, and no increase in Th17 cells, Th22 cells, or the makeup of the T cell subsets within the CD4 cluster. So is there a way that we can reduce the density of these BV type bacteria, in particular those basic bacteria, to reduce HIV risk without a surgical procedure? And to investigate that, Dr. Ronald Galawanga recently completed a randomized clinical trial where 125 men presenting to Entebbe General Hospital were randomized to either undergo immediate circumcision, which is the reason why they came to the hospital. That's 25 men. And then 4 additional groups were randomized to one of 4 different antimicrobial interventions. One was oral tinidazole, very similar to metronidazole that's used to treat bacterial vaginosis commonly in women, but just given for a 2-day course. And 3 topical interventions, topical flagyl, topical clindamycin, and topical hydrogen peroxide. The topical metronidazole and clindamycin are also treatments for bacterial vaginosis in women. Primary endpoint in this study was virus entry into foreskin CD4 T cells at the time of circumcision 4 weeks later. And the study is now complete. 116 out of 125 men completed follow-up, so were given the product, applied the product, and came back for circumcision 4 weeks later. And it's early to give you the final results. Unfortunately, shipping of our samples was dramatically delayed by the COVID pandemic, but I can give you initial results of the virus entry assays that were done in real time at the IAVI facility. And what you see from the APM trial, that's the Ultra Penile Microbiome trial, is that the control group has higher levels of pseudovirus entry than the tinidazole group. So we see a significant reduction in virus entry in those men who received tinidazole, again, the oral equivalent of metronidazole for bacterial vaginosis treatment, whereas we saw no impact on virus entry in any of the topical groups. I've lumped them together because they're really much of a muchness. And that may relate to CCL5 expression. So we see again within the tinidazole group, decreased CCL5 expression on foreskin targets, whereas we see higher levels both in the topical group and in the control group. We really hope to be able to present to you the microbiome and the cytokine data soon, and those samples have now been shipped, and we'll be able to run those assays. So just to summarize, foreskin is clearly linked to increased HIV risk. We think that a lot of that risk is driven by the microbiome, and in particular by these 6 bacterial species, these 6 basic species that are linked to both IL-8 levels and to an increased density of CD4 T cells, particularly within inner foreskin tissues. And it looks from the preliminary data as if we may have some success in changing immunology and HIV susceptibility by intervening on the microbiome without the need for surgical interventions. I want to acknowledge a lot of people who've helped out with this. Funding from the Canadian Institutes of Health Research, which is our national federal funding agency. My own group, particularly Sonia Hubner, who runs many of the cytokine assays in our lab. Dan Park and Cindy from George Washington. The Rakai group, there are too many people really to list, in particular Godfrey Kigosi, who's been amazing at helping to coordinate many of our studies. Aaron Tobian, Ron Gray, Maria Wawa from Hopkins, and most recently Brenda Oketch, who is the scientific director at IAVI Uganda. Vicky, I've underlined here because she ran many of our flow assays in that clinical trial. And of course, Jessica Proja from UWO, who ran much of the immunohistochemistry and all the previous immunology studies within my group. So thank you very much, everybody. And I hope there's now a way for you to give me some questions, but thank you for your interest.
Thanks, Rupert. That was a fascinating presentation, and there are a few surprises there, one with L. crispidus, and I'm sure we're going to get a lot of questions. So it's now my pleasure to introduce the next 2 short talks, and the first is by Kaitlin Marquis, and she's going to be talking about HIV replication, transmission, and the metabolome of the female reproductive tract. And then following that presentation, we have Brianna Jesaveluk from the Burnet Institute in Australia, and the title of her presentation is Lactic Acid Produced by an Optimal Vaginal Microbiota Promotes Cervicovaginal Epithelial Barrier Integrity, Implications for HIV Transmission. So we'll go to the first talk. Thank you.
Kaitlin Marquis. I'm a graduate student in Rick Bushman's lab at UPenn, and I'm here to talk to you today about my project on HIV replication, transmission, and the metabolome of the female reproductive tract. So I'm really interested in trying to understand how microbial-associated metabolites can modulate HIV replication in the vaginal tract. We know from work done by Duquan and others that women who are colonized by lactobacillus-deficient, highly diverse microbial communities, which are referred to as HDCs, are at a higher risk of acquiring HIV infection compared to women who are colonized by lactobacillus-dominant communities, or LDCs. And we know on the LDC side of things that the production of the metabolite lactic acid is really essential to establishing these protective effects of the community. But on the HDC side, we really don't have a good understanding of what metabolites are playing a role in this process. And part of the reason for that is because these communities are so heterogeneous in nature. Much of the existing literature has focused on the production of short-chain fatty acids, but I'm really interested in identifying sort of other metabolites that might play a crucial role in this process. And so with that said, these are the 3 questions I hope to address in the next 7 minutes. 1, are there conserved changes in the metabolome of cervicovaginal lavage fluid collected from women colonized by HDCs versus LDCs, despite the inherent polymicrobial nature of these communities? 2, could any of these metabolites directly affect HIV replication in target cells? And 3, are changes in the metabolome being driven by differences in the microbial metabolic machinery? And so in order to determine whether or not we can find any consistent changes in the metabolome, basically mined literature data to make a heat map where each different row of the heat map here is a different metabolomic study, and each column is a different metabolite that was measured and found to be significantly different in cervicovaginal lavage fluid between HDC and LDC colonized women. And what I want to focus on is sort of this core set of metabolites here that had really consistent changes in at least 3 out of the 4 different studies. And if you look at the makeup of this core set of metabolites, it's actually almost all amino acids, or they're metabolites that are produced from amino acid catabolism, with the exception of the lactate, inosine, and glycerol metabolites. And so then we were interested in trying to, determine whether or not any of these metabolites could actually be directly affecting HIV replication in target cells. So we developed a high-throughput screen based on the TZMBL reporter cell line that produces luciferase in response to increasing amounts of HIV virus replication. And so here's the workflow of the screen. On day 0, we plate the cells, and we added, I believe, over 500 metabolites from our metabolite library. And then we allow that to sit for 24 hours before infecting with a duotropic HIV. And 48 hours later, we conduct a luciferase assay to measure the production of light as a proxy for HIV virus replication. We conducted the screen in duplicate and were able to identify a number of putative enhancers and inhibitors of HIV virus replication. So then what I did was basically map the overlap of the results from the TZMBL screen and our metabolomic studies to identify the small number of metabolites here whose concentrations are different between HDC and LDC communities and affect HIV replication in our screen. And one of the metabolites that sort of caught our attention early on is this 2-hydroxyisovalerate molecule, which has been proposed as a biomarker for HDC communities as it's found in increased concentrations, and it increased HIV replication in our TZMBL screen. So we followed up on these results by treating primary CD4 T cells with this compound. On the left, I'm showing you differences in the amount of extracellular P24 of anti-CD3 stimulated donor CD4 T cells. So you can see that we found a dose-dependent increase in the amount of P24 in the supernatant of 2HV treated cells in a similar manner to our positive control sodium butyrate. And we also saw this when we took unstimulated CD4 T cells from 3 different donors and treated them with 2HV and infected them with the R5 tropic virus. So here we're using flow cytometry to quantify changes in the amount of HIV P24 positive cells. And you can see in orange, again, at higher concentrations of 2HV, we get an increase in infected cells, again, in a similar manner to our positive control sodium butyrate. And so with that said, what we've seen so far is differences in amino acid content between HDC and LDC communities, especially this 2HV molecule. So we were actually really interested in trying to determine whether or not this could be due to differences in microbial metabolic machinery. So we explored the number of amino acid biosynthesis genes between bacterial species that we can actually grow in the lab that have been isolated from either LDC or HDC women and are associated with HIV protection or acquisition. And actually what we found is that vaginal lactobacillus species tend to have much lower numbers of amino acid biosynthesis genes compared to their HDC and HIV associated counterparts. And furthermore, if you look particularly at the branched chain amino acid metabolism that produces the R2-hydroxyisovalerate metabolite of interest, you could see from our metabolomic study that you actually can see a decrease in the amount of this parental branched chain amino acids and a corresponding increase in the amount of catabolites produced by branched chain amino acid metabolism, suggesting this pathway might be important for regulating HIV replication in this setting. And furthermore, it was also sort of interesting to note that there's sort of an overlap with lactic acid metabolism, in that the LDHA enzyme is involved in branched chain amino acid catabolism. And so what we're working on now is actually developing a flux balance model of metabolic networks from personalized in silico reconstructions of vaginal microbiomes. So we can actually reconstruct individual microbiomes using 16S data, map them to a database that has done metabolic reconstructions, and we can actually model growth and metabolite cross-feeding among different communities and sort of identify metabolites that might be really important for this process. And our plan is to integrate the results we get from our dry lab efforts with our wet lab efforts, so we can continue to cross-reference the results from the dry lab and our high-throughput screen, and then follow up by determining HIV replication kinetics and immune cell phenotype of our infected cells. And even we want to get into measuring metabolites of interest in bacteriological culture media and vaginal fluid. So with that, I would like to thank everyone who's been involved in this project and for our funding. And thank you to the organizers for giving me the opportunity to speak to you today. With that, I will take any questions. Thank you.
Hello everyone. I would like to thank the conference organisers for the opportunity to speak today. HIV disproportionately affects women and adolescent girls in sub-Saharan Africa, where the majority of new HIV infections occur. In 2019, 30% of new infections in Eastern and Southern Africa were in females aged 15 to 24. HIV primarily establishes infection in women at the female reproductive tract epithelial barrier, which is composed of several discrete regions. These include the endocervix, which is a single layer of columnar epithelial cells, and the ectocervix and vagina, which consist of multi-layered stratified squamous epithelium. Epithelial cells are linked to one another by intercellular junctions. These junctions maintain epithelial barrier integrity and prevent the movement of foreign material and pathogens such as HIV between cells. However, epithelial junctions are modulated by the vaginal environment, and their function can be disrupted, such as by inflammation. The vaginal microbiome influences the environment of the lower female reproductive tract. An optimal microbiome is characterized by lactobacillus dominant colonization and is non-inflammatory. A non-optimal microbiome is one that is typically colonized by non-lactobacillus or diverse anaerobic microbial species and results in a pro-inflammatory state and bacterial vaginosis. A study by Gosman et al. Found that women with a non-optimal microbiome had a 4.4-fold increased risk of acquiring HIV relative to women with a non-iners lactobacillus-dominated microbiome. In sub-Saharan Africa, a large proportion of young women have a non-optimal microbiome, increasing their risk of HIV and other STIs. This highlights an important opportunity for an intervention strategy for the prevention of new HIV infections. A major metabolite of lactobacillus species is lactic acid. The majority of lactic acid in the female reproductive tract is produced by the vaginal microbiome, with a small proportion being produced by vaginal epithelial cells. Lactic acid is produced as D and L isoforms. The ratio of D to L lactic acid produced varies between lactobacillus strains, and the isoforms have been shown in some studies to have differential effects. Protonated lactic acid has been shown in vitro to mediate a number of protective effects for vaginal health at pH less than 4.5. At a neutral pH, the lactate anion predominates. Lactic acid maintains a low vaginal pH, which is important for limiting the growth of opportunistic BV-associated bacteria. In addition, lactic acid inactivates HIV and a number of other STI pathogens directly. Our laboratory has also characterized the anti-inflammatory and immunomodulatory effects of lactic acid. We hypothesized that lactic acid also impacts the vaginal environment by modulating epithelial barrier integrity and tight junction protein expression. To evaluate this, we used an ectocervical cell line called ECT1 and seeded cells on semi-permeable transwall supports to establish a monolayer. Cells were treated apically for 1 hour with 0.3% L or D lactic acid at pH 3.9 or 7, as well as with media adjusted to pH 3.9 with HCl. Transepithelial electrical resistance, or TEER, was measured prior to and 24 hours after treatment as a measure of barrier integrity. We found that TEER after 24 hours was increased significantly by treatment with 0.3% L or D lactic acid at pH 3.9, but not at a neutral pH. This effect was acid independent, as media acidified with HCl did not significantly increase TEER. We then performed RNA sequencing of ECT cells in 4 independent experiments. Cells were grown and treated in transwells for 1 hour. The treatment groups were untreated cells, L or D lactic acid treated, or HCl treated. 4 hours after treatment, RNA was extracted and cDNA was synthesized for NGS. Quality control validation was performed and differential gene expression was analyzed using the platform dGUST. Gene ontology enrichment analysis was analyzed using the Gorilla online tool to perform functional analysis. Findings were validated using qPCR for gene expression and SDS page and Western blot for protein detection. Gene ontology enrichment analysis of cellular components showed enrichment of a number of gene ontologies associated with intercellular junctions. Of the 14 enriched gene ontologies for cellular components, 6 were associated with barrier function. RNA-seq analysis showed a large number of differentially expressed genes following treatment with lactic acid relative to untreated cells. Over 148 genes were found to be differentially expressed relative to treatment with HCl. We observed no differentially expressed genes between L and D lactic acid groups. We analyzed expression of a panel of tight junction genes and identified at least 15 that were significantly differentially expressed. We then selected 5 notable tight junction genes for further analysis. Differential gene expression analysis was measured by qPCR to validate the findings from the RNA-seq analysis. Differential gene expression was similar across both methods, with Claudin-1 and Claudin-4 showing the greatest fold change in expression relative to untreated cells. Analysis of differential expression of tight junction proteins using Western blot analysis showed that Claudin-1 was significantly upregulated after lactic acid treatment relative to untreated cells. Claudin-4 was additionally found to be significantly upregulated after lactic acid treatment, but also by HCl treatment alone. In conclusion, lactic acid increases barrier integrity of cervicovaginal epithelial cells by modulating expression of tight junction genes and proteins, which may contribute to the protective effects of a lactobacillus-dominant microbiome against HIV. Lactic acid could potentially be used in a preventative strategy in young women to reduce the risk of HIV acquisition. I would like to acknowledge my PhD supervisory team, Gilda, Anna and Josh, and everyone involved in conducting this study, particularly David Delgado Diaz, who performed much of the experimental work shown, and Joshua Hayward, who led the RNA-seq analysis. Thank you.
Thank you. I'd like to thank Bree and Kaitlin and all these speakers in this session for excellent presentations, which have that common thread of informing strategies to harness the genital microbiome to prevent the sexual transmission of HIV. So as expected, there's a lot of questions. So I'll start with the first question, and that's to Laurel. From Ron Coleman to Laurel. You mentioned plans for cytokine analysis. Do you have any initial data? The question would be, The question being that, whether therapeutic repopulation with normal type microbiome drives to low inflammatory state, which then presumably drive to decreased HIV infection, Laurel.
I know that I think it's Doug Kwong's group had shown that women who transitioned from BV to lactobacillus did have decrease in inflammatory cytokines, but due to COVID, we haven't been able to analyze those data, but Rupert will get those samples and be working on that at some point.
Okay, we look forward to seeing the data. Okay, the next question's from Lindy Masson, this is to Laurel again. Did you look at whether women with recurrent BV were more likely to have new or multiple sexual partners?
So they did collect data on sexual partners, but that's something we're starting to tease out now as we look more deeply into the data. So I don't have any information on that at this point.
Next question again to you, Laurel. For the women who LACTIN-V did stop recurrence of BV, what would be the next step to treat them?
So we're looking at whether we could evaluate women with a diagnostic or some sort of test at 4 weeks. I know that Becton Dickinson came out with a nice BV test recently. And if women could test themselves, they might continue therapy with LACTIN-V longer, because some of the women colonized and then their levels dropped when the dosing stopped. And so for those women, continued dosing might be useful.
Yeah, and I might just follow on from that question. Did you follow on beyond 48 weeks to see whether those women that were colonized with LACTIN-V remain colonized?
No, the last time point was at 3 months post dosing, which was at 24 weeks.
Okay, all right. So we have a question from Cross Cromati again to you, Laurel. Non-white women tend to have higher prevalence of inners rather than crispatus as their natural state, especially in Africa. Could this be the reason why some women did not stay in the crispatus state? And were these data stratified by race?
The data were looked at by race. We did see decreases in BV in both white women and in women of African American and Hispanic. White women did do a little bit better in the study. We don't have statistical significance on these data, but we're looking further at that.
Great. Another one to you. I'm just going through these chronologically. This one's from Shane. Since it's known that white women tend to naturally have higher amounts of lactobacillus species other than crispatus, why was a L. crispatus strain selected? And also, do you foresee that these women will then have L. crispatus as the dominant vaginal species in the long term?
Right. So Lactobacillus crispatus was selected for a number of reasons. It produces both D and L lactic acid. It has a long safety record, the particular crispatus strain we're using. And crispatus is the lactobacillus that's most associated with vaginal health and lower inflammation. Now, that being said, a second generation product might include a different lactobacillus or a combination of lactobacilli to try to cover the basis for all women. Did I answer everything there?
Yeah, more or less. Okay. Can you see the question there?
Would women continue to be colonized with crispatus? If what we saw is women who were colonized continued to be colonized, but we did see women drop off. So. Okay.
All right. So the next question goes to Rupert, and this is from Laurel. Fascinating that Lactobacillus crispatus actually increased IL-8, and I thought that was fascinating too, while Staph did not. Was there a Staph aureus or was this Staph aureus or Staph epi? Rupert, if you're there.
I'm not able to give you the breakdown in terms of species on that yet. I certainly think it's likely to be Staph epi. I mean, these are skin type bacteria, so I think the great majority are going to be the more common skin staphylococcal species. So that would be epidermidis. And I do see that you were asking Laurel as well. Oh, maybe I shouldn't presuppose. Gilda, you.
No, you can go ahead.
So there was another question from Laurel about the crispr. So Laurel had also said here, since I'm answering Laurel's questions, how would you resolve the findings with Craig Cohen's findings that BV was associated with more transmission to partners? And it makes me, as you can see from my talk, I'm not very disciplined at all. I'm always presenting preliminary results that I find exciting. And so we are still doing some of the analysis with many of the studies. There's another study that I'm really interested in, which is the sex study, where we have partners have sex and then very carefully monitor immunology and the microbiome after having sex. And what we see, nobody's really looked at that before in terms of penile immunology and penile microbiome. So you see really dramatic changes in penile cytokines after sex, kind of as you would expect. So you see really dramatic increases in inflammatory cytokines that persist for at least 8 hours. They've generally resolved by 48 hours, but they persist for at least 8 hours after sex. In an explant model, if I put cytokines on a foreskin explant, that substantially increases HIV transmission. And so we think that likely what's happening is that essentially after sex, the penis is getting coated with female genital tract secretions that are high in inflammatory cytokines, and that may be associated with HIV transmission, so that a woman who has BV has more inflammatory cytokines, so that may be what drives that transmission. The crispr question is very interesting. Cindy has those samples from us. We do not yet have any data whatsoever on the time course of microbiome changes after sex. So I can't tell you how long, crispr would hang around on a penis after sex. We just don't have those data yet.
Thanks, Rupert. Another one here from Jim. Several years ago, there was a hypothesis that increasing penile cleanliness, washing, would reduce transmission. Your antibiotic data seems to contradict this. Any comments?
Sure. There was actually, there was a study that came from Nairobi that I was collaborating with the group that was doing it. So we used to joke about it, called the Willy Wash study is what we called it. I forget what they were cleansing the penis with. Essentially, it was a trial where they were looking at, it might have been chlorhexidine wash. I believe it was chlorhexidine wash in women who had bacterial vaginosis to see whether you could reduce, basically if the men were randomized to either use chlorhexidine or not, to see whether that could reduce BV recurrence in the female, in the woman who was the index case with BV. And what they saw was that men who used chlorhexidine actually, there was increased risk of BV recurrence in their female partners. So I think that, altering the microbiome, things that result in disruption of the microbiome acutely, may or may not actually be good in terms of inflammation. So I think we need to know much more about what dynamic changes in the microbiome do in terms of inflammation. And that's something that I can't really present unfortunately from our penile, the APM trial, the Alter Penile Microbiome trial. We don't yet have the microbiome data at all from that because of problems shipping to Cindy during COVID. So I can't tell you which of our interventions actually changed the microbiome, which I think cuts off a few of the later questions, Gilda.
Okay, all right. Well, I'll still ask them. So Adam Bergener, g'day, Adam. Great to see your talk yesterday. So he says, really great talk, Rupert and data. Could the antibiotic treatment have other effects on the epithelium that might also explain the lower viral uptake infection in your pseudovirus assays?
It's a great question. We do think that, there are two potentially independent, but often they go together because inflammation can drive both of them, processes that are linked to HIV susceptibility. That's epithelial breakdown, and that is recruitment of target cells. So, inflammation leads to both of those, but they're actually two separate processes. We were able to look at an epithelial biomarker. So we're going to be measuring a few epithelial biomarkers, but we didn't actually have epithelial integrity through immunohistochemistry as one of our readouts. So the immunology readout that we have, the virus entry, is digested tissue. So we take the foreskin tissue, we digest it, and we look at virus entry into that tissue. So I can't really, that's independent of anything that might have happened to the epithelium. So the changes in the epithelium can't really explain the virus entry because we've actually taken out the tissue. We're not putting virus on the epithelium and watching it go into cells. We're digesting the tissue, taking out the cells, and then looking at virus entry. There may, in addition to those T cell changes, there could be epithelial changes. We'll get a clue for that from the epithelial biomarker data, the ECAD-Heron and MMP9 data, but we won't be looking at it directly in terms of immunohistochemistry, at least not initially.
Okay, so Rupert, you again. Lindy's got a question for you. Was penile L. crispatus relative abundance positively associated with the other bacteria that were linked to IL-8? So that's the first question.
So, crispatus is really interesting. And it wasn't really it's something that I'm particularly interested in because I also do work in the female genital tract. It's not necessarily something that the penile microbiome group are as interested in. They seem puzzled sometimes by my great excitement around lactobacilli species and Lactobacillus crispatus. It's not found in all individuals. In again, another independent study that we're doing, it does appear to be linked to sexual activity. So one possibility is that the answer is it's not found in most men. It's not part of the sort of normal foreskin microbiome in the same way that those other species are that I'm talking about. So it didn't really fall into the map in quite the same way. When it was present, it was associated with more inflammation, more IL-8. And it's possible that it may relate to recent sexual activity. And that's why I'm very interested from the couple study that we're doing to see how long Lactobacillus crispatus or inners, depending on what the predominant Lactobacillus species is in the female partner's genital tract, how long those lactobacilli hang around after sex, I think is going to be very interesting to look at. And so, that we'll hopefully have an answer that can expand on that soon.
Yeah, I'd wonder about that too, because I'd be wondering what the energy source is. There's no glycogen, is there, in the penis for the lactobacillus to propagate? And so just continuing with Lindy's question, was L. crispatus still positively independently associated with IL-8 if you adjusted for the relative abundance of other bacteria such as Prevotella?
Yeah, as I say, because it wasn't one of the predominant bacterial species present in most men, I don't think we've really chased that. So I can't give you an answer to that, Lindy, but it's something that would be fun to look at. It's true. I can get Dan and Cindy to have a look at that. And certainly, I think if there's a lot of interest in the field in that lactobacillus question, it might be interesting to do a whole separate analysis around lactobacilli within those cohorts, which at the moment we haven't really done in the analyses we've done.
Okay, and here's another question for you, Rupert, from across
of the younger speakers.
Yeah, I want to make sure that the young speakers get some questions for theirs too, Gilda.
I see several questions.
I'll just finish, I'll get to the young one. So what are your thoughts on the long-term impact of the use of these antibiotics on the mucosal environment of the penis, as the microbiome is natural and plays a big role in the mucosal states?
Well, that's a super that's a super good question. This was designed as a pilot study. We didn't really know what the effect would be. I still cannot tell you what the effect was on the microbiome. We still don't actually have the microbiome. We only have data for that primary endpoint. And so I think it's unfortunately a little bit premature for me to be talking about what might be the effect on, of microbiome changes on epithelial integrity in the long term, because we don't even know to what degree we change the microbiome yet with our intervention. So we may be able to look at that and correlate it specifically with epithelial biomarkers. We do have tissues cryopreserved, so there will be future opportunities, I think, to actually look directly at immunohistochemistry, immunohistochemistry for markers of epithelial integrity. And that may give us a hint as to what the long-term implications might be.
Okay, so I'm going to move to the questions for our graduate students. So Kaitlin, this one's from Satya. Excellent presentation. Is it possible that these metabolites are working at the epigenetic level in immune cells?
Yeah, sure. So I can't say I've looked too much into specific mechanisms yet, because we have certainly a lot of questions that we need to answer before we get to that level. But definitely my inclusion of, butyrate, for example, in there as a positive control, we know that works at the epigenetic level. And there's certainly the potential for other metabolites that come out of our screen to also function at an epigenetic level. But I don't think, at least for the branched chain amino acid catabolites and the ones that we're particularly focused on, now are working sort of at that level. That seems to be more through metabolic reprogramming through mTOR signaling.
Thank you, Kaitlin. So another question, this is from Satya for Brianna. Bree, you've got your mic here, that's great. Great job, Bree. Have you looked at any signaling pathways that get activated due to lactic acid treatment?
Thanks for the question, Satya. We will be doing an analysis of transcription factors in our transcriptomics data to attempt to identify signaling pathways, but unfortunately we haven't conducted this analysis yet, so I don't have any insights that I can share today. We're also interested in identifying potential receptors for lactic acid. We hypothesize that there may be an internal intracellular receptor, given that lactic acid is an uncharged molecule and it should be able to pass through the cell lipid bilayer without engaging with a cell surface receptor.
Thanks, Bree. Well done. It's 2:47 a.m. in the morning in Melbourne, so you're looking bright. Okay, the next question is for Kaitlin. Awesome work. This is from Catherine Lozapone. What do you think the mechanism is of BCAA catabolism product promoting HIV infection? Do you think it's through immune modulation or something else?
Great question. Yeah, this is what I wrote my thesis proposal on. So what's actually really interesting is that there's been a lot of sort of recent interest in branched chain amino acid metabolism and immune modulation. So we know that actually in CD4 T cells that there is a cytosolic isoform of the sort of terminal enzyme involved in branched chain amino acid catabolism that's not expressed in other cell types. It's very tissue restrictive. And we know that that particular isoform is really important for CD4 T cells to be able to basically metabolic reprogram themselves to like become more activated and produce cytokines. So there's actually studies showing how depleting these types of amino acids can affect CD4 T cell phenotype, cytokine production, and activation status. So yeah, my guess is that it probably is regulating HIV replication by sort of regulating that sort of activation level of the CD4 T cells. And that's one of the things I'm personally very interested in measuring alongside sort of our studies of just what it's doing to the amount of HIV in supernate. So yeah.
We've still got time for more questions. Bree, if you're there. This is a question from Cross Cromarty. So since there are no differences in tier between HCl and the LA isoforms, what are your thoughts on the protective effect of lactic acid compared to HCl? So I think you did show a difference.
Yeah, thanks for the question. I'm sorry if it wasn't clear, but we did actually observe a difference between HCl and the LA isoforms in tier. So the lactic acid increase in tier was statistically significant, but there was no significant increase in tier with HCl treatment.
Okay, thank you. And I think there was another one I missed, and that was for Laurel, and it was about the dosing of LACTIN-V, what form it was and how often it's dosed, which I think you covered in your talk, but if you can
it's dosed as a powder delivered in a vaginal applicator, and it was dosed for 5 consecutive days and then twice weekly for 10 weeks. We're also looking at other potential forms in the future, perhaps fast dissolving tablets or direct compression tablets.
Okay, yeah, and that was for Delia. Thanks, Delia. There's another question for you, Laurel. It's from Bhavna. Did you look at effect of LACTIN-V on recurrent BV in women on contraceptives, which is an interesting question.
We did ask for contraceptive use. Some women were on the copper IUD, some were taking DMPA, some were on oral contraceptives. And right now we haven't been able to tease anything out because there were so many variables at this point. But yeah, I know others have looked at that, and there was a talk yesterday clearly from Adam on different contraceptives, at least as far as what they did to the microbiome.
All right. Okay. So I've got Nikki Clash on the line. G'day, Nikki. This is to Kaitlin. Have you assessed whether mucosal metabolic alterations are associated with systemic inflammatory changes?
Yeah, that's another good question. So I haven't done that personally, but I've done a lot of reading sort of on that topic. And I can tell you, at least for the branched chain amino acids, that there's a lot of literature evidence to suggest that's tied into systemic inflammation. And actually, we know from studies from people who have metabolic disorders, such as maple syrup urine disease, which is a disease we actually see a lot of these branched chain amino acid catabolites in blood plasma. You have also high levels of systemic tissue inflammation in those settings too. So I can't speak for this setting in particular, but it certainly seems like there is the case that could be what's happening.
Okay, thank you. So Laurel, I'm going to ask a question. So with LACTIN-V, are you considering, or is there any consideration for using LACTIN-V to prevent spontaneous preterm birth, in particularly the infectious causes?
We actually have a clinical trial in progress, yes.
Okay, excellent. And I did mention in the introduction Mucocept HIV. I was just wondering, where that is in the pipeline.
Right. So we've recently received a phase 2 SBIR to reformulate because we had issues with the formulation, not with the lactobacillus itself, but with the placebo part of the formulation. So we're reformulating that in a lower osmolality tablet to be, or powder to be used. We'll test it again in an animal model, and then hopefully we can move into humans with that if the animal model shows no inflammation.
Great, thank you. I'm going to now hand over
I want to just quickly say the two younger speakers were just excellent. I really appreciated both their talks.
Thank you. I'm going to hand over now to Jim to close the session.
Okay, so thank you, Gilda. Thank you, our speakers. Some very interesting, very provocative discussions from our older, more experienced speakers, and our two younger speakers, who I think gives us the future of where microbiome may be going and the things we need to think about for the future microbiome conferences. So with that, I will end this session saying thank you very much for everybody for joining. And I think we are now moving into an abstract session.